on CMDE score and M score. CMDE score is our method for estimating binding affinities. It's an empirical method for estimating protein-protein and protein-peptide binding affinities. And CMDM score is our molecular mechanics-based method for estimating protein-protein and protein affinities.
Did he just shut his mic off?
Without it, oh, there we go. So this is some recent testing of CMDE score. So you can't succeed in structure-based design without some kind of a method for estimating binding affinities. And it has to be fast, and it has to be accurate. It's arguably the key to successful structure-based design of any kind, whether it's proteins, peptides, or small molecules. And what was nice is that recently, two groups described a protein-protein binding affinity benchmark in the literature. So this provided us with an opportunity to really do some rigorous and blind testing of this empirical scoring methodology that we had developed. And to make a long story short, we got what we think are encouraging results. So this is the original work. And so it's for estimating binding affinities. That's actually the expression. It's a 6-term expression. And it was weighted or parameterized on a training set. And those are the results from the initial test set. So it was a 35-member protein-protein and protein-peptide test set. And we got very, very nice results, as you can see. And this protein-protein benchmark that was recently described in the literature, this isn't a benchmark that we put together. This is a benchmark set that independent groups put together. So it provided us, like I said, with a really nice opportunity to rigorously test CMDE score and its accuracy in a blinded manner. And as you go from the top left to the right, you better approximate the rigid body assumption. Okay? And as you go from the top left down, okay, you get higher quality binding affinity data. Okay? So in the bottom right, that would be the most relevant test set. Okay? The other ones have some meaning, but the bottom right has a lot of meaning. And we got some very nice results there. So we're really happy with that, and we're really encouraged by that. And just to put this into context, you can use CMDM score, E score, to estimate these protein-protein and protein-peptide binding affinities in say, 15 seconds, 20 seconds, 30 seconds. So you can estimate binding affinities in a high throughput fashion, which is nice. Then we have CMDM score, and this is a molecular mechanics base, so some would say it's a more rigorous approach to estimating binding affinities than E-score, which is an empirical approach. It's basically a molecular mechanics expression, and it's an implicit solvent model. There's very little in the way of parameterization, very little in the way of training. And the data so far looks pretty encouraging. It looks pretty good. And this is important to note about M-score. It's a molecular mechanics approach, like I said, but it's basing the binding affinity calculations off of single structures. So not like a molecular dynamics simulation or a Monte Carlo simulation or something like that. It's a single structure. So it's fast. It's very fast, actually. And these are the results. So to the left, those were the original results. So I forgot to, on the y-axis would be experimental binding affinity, and on the x-axis would be predicted binding affinity. So that was the first test set was to the left. That's from about a year ago. And then to the right, that's more recent data. So once again, we're seeing what we think, at least, are very nice results, and we're really encouraged by the results. So with CMDE score and CMDM score, we have kind of a hierarchical way to quickly and rapidly estimate protein-protein and protein-peptide binding affinities. And like I said, that's the key to successful structure-based drug design, structure-based peptide drug design in this case. Now this is some data that we're really excited about. And we've spent the last year trying to develop a de novo method for predicting peptide structures from sequence. So we can now announce, I guess, CMD peptide. And so this module, this program, can be used to predict 3D peptide structures from 1D peptide sequences. And it requires minimal information. It's physics-based and it's fast. It uses what we call a multiple start Monte Carlo algorithm to generate conformations, peptide conformations. And then the conformations are scored using a physics-based scoring function. And once again, this is important, I can't emphasize this enough, there's very little in the way of training. It's a pretty pure method, it's a robust method. And this is the basic workflow. So you start with an input sequence, so that would be the peptide sequence. And then what the program does is it builds multiple 3D start structures, okay? And then it takes those start structures and it processes them through what we call our Monte Carlo gauntlet. And then that results in about 360 peptide conformers, okay? And then those 360 peptide conformers are subject to the scoring protocol, which is basically a physics-based scoring function. And then the final result is going to be about 20 peptide conformers that are enriched, and what the data seems to show, enriched with native-like peptides. And then you can use those peptides, you can subject them to further analysis, or you can use them for docking to do ADME predictions and things like that. And here are some of the results. And once again, we didn't put this test set together. Somebody else put this test set together. So we're testing our method on a test set that was put together by another group independently of us. And we actually have a poster on this. So if anybody's interested, we'll be showing the poster tomorrow. There's a lot more information and a lot more data. But basically, this is, I think it's 12 of the peptides. And the all-important trip cage peptide is included in there. And the reason I mentioned the trip cage peptide is a lot of people who do computer or computational peptide structure prediction, they like to benchmark against the trip cage peptide. And there's a slide on that. And you can see you have a variety of lengths. I think it's up to 24 amino acids at this point. And then you can see for every single peptide, we're able to sample a native-like peptide conformation in the top 360 conformers. And then using the physics-based scoring function, in pretty much every single case with one exception, at least with this data, we were able to select out a native-like peptide conformation. So we're happy with these results. We're encouraged by these results. And like I said, we're presenting a poster on this if anybody wants more information or wants to talk about it. Just to put these results into perspective, I had mentioned the trip cage peptide. A lot of groups who try and do peptide structure prediction, they try and benchmark against the trip cage peptide. So we decided to compare our prediction. The 1 angstrom result there, that's the top ranked prediction. That's the top scoring prediction. So we decided to test it against Rosetta. There's a method called Quark, which is an ab initio protein structure, peptide structure prediction method. And then there was a paper published by, where the people who published the paper, it was a very sophisticated simulation. CHARM, generalized Born solvent accessible surface area solvent model, and what's called replica exchange molecular dynamics simulation. So much more computationally expensive than CMD peptide. And as you can see, our results compare very nicely with the state of the art. Now what I want to talk about is kind of putting it all together. When we put all these modules together, we try and actually do something like peptide hit identification for a partner company. So in this case, this story that I'm about to tell, the partner company is a New Haven, Connecticut-based company, small biotech company, L-Squared Diagnostics. And they were basically interested in novel peptide inhibitors of dengue virus and all 4 dengue serotypes, so dengue virus 1, 2, 3, 4, and West Nile virus. So they wanted a single novel peptide that would exhibit inhibitory activity against all 4 dengue serotypes and against West Nile virus. They specifically wanted peptides to block this all-important stem peptide envelope protein interaction. At the time, the gold standard peptide inhibitor was a 30 residue soluble stem peptide, truncated stem peptide. They had experienced 2 years of failure using, I guess what you might call wet discovery methods or more conventional discovery methods. So what we ended up doing for them is we used CMD Inventus to generate and then screen 480,000 unique peptide sequences against the stem peptide target. And when it was all said and done, it's a small company. They don't have a big budget, so they didn't have much of an appetite for synthesizing lots of peptides. So we recommended that they synthesize about 27 peptides, and they actually got 5 nice hits from those 27 peptides that they synthesized. And this provides a little bit more detail. This project really challenged the software because the first thing, we didn't have a good crystal structure available to us, the envelope protein. So the first thing we had to do was we actually had to model in the missing stem peptide. We had to model that in. That's what I mean by it challenged the software. So we were doing structure-based design on a model as opposed to structure-based design on a high-quality, high-resolution crystal structure. So that's the first step there. And then in the second step, we used CMD scaffold to select out a peptide scaffold, and then we used our de novo peptide ligand design program, CMD Design, to basically generate the 480,000 peptides on that scaffold. And then we estimated the binding affinities using M-score and E-score. Then they did the testing, and those are the results. And like I said, it was they ended up synthesizing about 27 peptides. No, it was 27 peptides. They were happy with 5 of the hits. Something we were encouraged by is the confirmed hits had lower binding affinities, predicted binding affinities, than the misses. Okay, so we were encouraged by that. Subsequent testing showed that they were indeed broad spectrum, so they had good inhibitory activity against all 4 dengue serotypes and against West Nile virus. Activity testing against a panel of unrelated viruses, they exhibited no activity, so it's reasonable to conclude that they're more or less specific to flaviviruses. And they did a toxicity assay, and they were able to show that they're non-cytotoxic as well. So that just summarizes basically everything I just said. So this is just to emphasize the final point as far as the business model goes. We're not trying to position ourselves as a software company or as a fully integrated drug discovery company. Instead, we're trying to position ourselves as a platform company. And we're actively seeking out peptide drug discovery collaborations with biotech companies, pharmaceutical companies, and academic labs as well. So if you agree with that, come and talk to me, I guess. Thank you very much.
Thank you, Joe. I'm sorry I forgot CMD.
Intuitive way to see if that's working is to look at the top 20 and see if there's a native-like peptide in there. And there was for the most part. Another way to do it is to calculate the correlation between the score and the root mean square deviation. So the higher the correlation, the better job the scoring function is doing selecting out native-like peptides. And there was quite a range, as you could see. But for the most part, I think it was better than 0.5. Any other questions?
Questions for Joe? Thanks again, Joe.
Yeah, thank you.